All right, welcome to today's lecture, and we're going to be covering the first learning target in topic 1.3 from the College Board course outline for AP Psychology version 1. Specifically, we're going to be talking about experimental design and variables. The lecture itself was written and designed by me, Chris Gall. As you're watching this video, as always, feel free to pause or to rewatch as needed to go ahead and complete your notes. So our learning target for the lecture was brought to you by the numbers 1 and 3, and is that scholars can identify independent, dependent, confounding, and control variables in experimental designs. In English, you need to not only understand what these words mean, but be able to recognize them if you were given a scenario, or in the case of confounding and control variables, to be able to identify which ones might be in play. Okay. Luckily, a lot of this information should already be pretty familiar to you from your math and science classes as you're going through it, so hopefully you'll find this a quick and easy topic to master. With that being said, let's go ahead and just dig straight in. Okay, so when I was learning this stuff, like 300 years ago, plus or minus, I learned that independent variables are what is not being measured during the experiment. In other words, if the scientists are changing it, it's the independent variable. Okay. According to the APA, and I stole this definition and all of the other ones for this lecture from their online dictionary at dictionary.apa.org, independent variables are the variables in an experiment that are specifically manipulated or observed to occur before the dependent or outcome variable in order to assess its effect or influence. Okay. So let me give you an example real quick. In a psychology experiment, rats are being run through a maze. The psychologists are attempting to determine whether or not the rats can be taught to run through the maze quicker if they are fed a snack at the end of the maze. Okay. What's the independent variable in this experiment? Go ahead and pause it for a second, see if you can figure that out. To decide if you are correct, ask what's being measured and what's being changed. If it's being changed, then that's the independent variable. In this case, what's being changed is whether or not there's food at the end of the maze. So since it's being changed, that's the independent variable. Okay. In contrast to independent variables, which change across the course of the experiment, dependent variables do not change. Instead of changing, they're what is measured. Okay. So scientists are not directly changing the dependent variable. They're measuring it. So the question being posed is really how the independent variable changes what happens to the dependent variable. Okay. According to the APA, the dependent variable is the outcome that is observed to occur to change after the occurrence or variation of the independent variable in an experiment, or the effect that one wants to predict or explain in correlational research. Okay. So to determine what the dependent variable is, ask yourself what is being measured in the scenario provided. That will tell you what the dependent variable is. Okay. So in our experiment from the last slide with the rats running through the maze and the question being whether or not food influences how quickly they run the maze, the independent variable is the food. Okay. The dependent variable, what is being measured, is how quickly the rats run the maze. That is what we're actually measuring. Make sense? To confound means to confuse. Okay. If you know that, then it makes it easy to figure out and remember what confounding variables are. And these are variables that also influence the outcome. Okay, so they're kind of like independent variables in that they're going to influence the outcome. But the trick is that they confuse that outcome. So according to the APA, confounding variables make it impossible to differentiate the independent variable's effects in isolation from its effects in conjunction with other variables. In other words, confounding variables also confuse the outcome and are difficult or impossible to separate out from the effects of the independent variable. So to determine if a variable might be confounding the outcome, ask yourself if you could get an impact on the dependent variable by changing that confounding variable. Okay. In our scenario from before with the rats, how recently the rats were fed might be a confounding variable. Okay. If they're fed just ahead of running the maze, they're less likely to run quickly to get food once they know that food is what's on offer. If, on the other hand, the rats haven't eaten in a day, they will be hungry and will run the maze quicker, again, once they realize that food is on offer. In another example, if the mice are in light or dark, that might influence their speed through the maze. So it's going to confound or confuse it. Okay. Wherever they can, scientists try to control for confounding variables. In the previous scenario, they might control for how recently the rats were fed by being sure to feed them all at the same time ahead of when they run the maze. For the light or the dark, they may use the same lamp to light the maze so that the rats have the same amount of light, or do so in a room that is otherwise unlighted, otherwise unlighted, or similarly lighted each time the rats run it. So I spent the last slide talking about confounding variables, and this one is about control variables. And the big difference between the two is whether or not scientists try to ensure that they remain unchanged. That is to say that they are controlled. Okay. The APA says that control variables are variables that are considered to have an effect on the response measured in the study, but are not of particular interest to the researcher. To remove its effects, a control variable may be held at a constant level during the experiment, like feeding the rats at the same time compared to when they run the maze, or always lighting the maze in the same light, or managed by statistical means, for example, with a partial correlation. And partial correlations really just mean that scientists run some statistics and they say X amount of the change that you're seeing is a result of Y variable. Okay, so it's a way of saying we know that there are things that are confounding it, and here's how much they're actually confounding it. Okay, if a variable affects the outcome and is not controlled, then it's confounding. If it is controlled, then it becomes a controlled variable. Whenever possible, scientists try to identify all of the possible confounding variables and convert them into controlled variables with the procedures that they define. These are closely related concepts and tricky. To determine which one you were looking at, ask yourself if the scientist accounted for the effects of the variable. If they did, then it's a controlled variable. If the scientist did not, and it affects the outcome, then it's a confounding variable. Make sense? There we go. With this lecture, we are now beginning to dig into the experimental method. Hopefully, at least the idea of the dependent, independent, and control variables were all familiar to you from previous science classes, as well as hopefully from your math classes and at least dependent and independent variables. Okay. The one term you're likely not to have come across elsewhere is a confounding variable. So to be successful today, you should be able to identify and define what independent variables are, dependent variables, confounding variables, and control variables. Okay. To tell if you can do this, try reading an experimental procedure and just ask yourself, what are the independent, dependent, and control variables, right? So what's being changed, what's being measured, and what's being accounted for. Then ask yourself if you can identify any confounding variables. Okay. So what could affect the outcome but isn't being accounted for in the procedures? If you can, then if you can identify confounding variables, then the experiment was not as well designed as it probably should have been. If you cannot, then they have designed a really good experiment that should measure exactly what it was designed to measure. If you've got questions, go ahead and bring them into class and I'll answer them there. Otherwise, I will see you the next time we meet in class. Until then, live long and prosper.